I'm Joan Miller. I'm the chair of the Harvard Ophthalmology Department and chief of ophthalmology at Mass Eye and Ear and Mass General. I've been involved in macular degeneration research since the early 90s when I finished my fellowship training in retina. And at that time, we really had virtually nothing to do for macular degeneration except laser photocoagulation, which scarred the retina. So it was not great for patients and not fun for the doctors. And it was a great incentive to try to find something better. And I was lucky enough to be part of groups that helped develop the two mainstays of therapy for wet macular degeneration, which we have today. Photodynamic therapy, which was first, and then the work to block VEGF, or vascular endothelial growth factor, which has led to the drugs that we inject now pretty routinely. But clearly, where we need to be now is to find a treatment for the earlier form or intermediate macular degeneration, the dry forms, so that we can treat patients before there's ever any vision loss. And there's a lot of great work that's been done. I think we have a lot of the building blocks that we need to understand this, but it's about sort of putting them together in the right way that we can really find a good path forward. We have a great group working on macular degeneration at Mass Eye and Ear, and I'm excited to be part of that. Some of the things that we're working on now include trying to develop biomarkers for that earlier stage. And one of the areas we're working in is called metabolomics, and some of that was presented at the symposium this weekend. And metabolomics is one of these new omics where we study small molecules that are the metabolites that can be in your bloodstream or in urine. And what we try to do is to measure those and see if you can see differences between people with macular degeneration and those who don't have disease. And then look at different stages of macular degeneration and see if there are differences there. And our preliminary work is quite exciting. It looks like there are differences, and we're continuing to explore that. That work is really led by Diba Hussain in our group, and she's been doing a great job. We ultimately hope that we will have a biomarker where you can both identify patients with macular degeneration, but perhaps more importantly, provide prognosis as to who's going to progress to different stages, and ideally really come up with a personalized medicine approach where you can measure this biomarker and then tell a patient this is the drug treatment that you need to prevent progression. So that's where we're headed. We've got a ways to go still. I also work with Demetrios Vavas, who's a member of that macular degeneration group, and one of the areas that we have been working in for a number of years is called neuroprotection, where you're trying to prevent the retinal cells from dying, which ultimately is what causes vision loss and macular degeneration and other conditions. And we have some exciting work in different models that we're hoping to get to patients, and I think that will be another real added advantage, both hopefully over the long term, even for the early stages, but first to really improve our results with the wet or neovascular form, and also for geographic atrophy, which we really have no treatment for at the moment.
Are there particular biomarkers that are being looked at in AMD progression?
So I think there's a lot of interesting work going on in biomarkers, and they can be different things. One of them is actually looking at the imaging that we have now. We have so many great tools to image the retina, and there are new ones that seem to be being developed all the time. And we can actually look at the imaging probably using artificial intelligence or deep learning to detect patterns that we actually as humans can't see. So you might have an imaging biomarker that indicates that this person, has a greater risk for progressing onto the atrophic form of macular degeneration, and we need to, again, treat them more rapidly and with drug Y once we get it. With patients who present with a wet form of macular degeneration, it might be that the imaging biomarker could say, well, this person's going to need a lot of treatment, or this other person may not need so much. And that was presented a little bit this morning by Tony Adamus. Other kinds of biomarkers are things that we can measure in the blood or plasma or in the urine, and it would be a simple kind of test that you look for a particular pattern of metabolites, and it requires it sort of would be like a chip technology where you get the sample and then process it very quickly to get an answer, again hoping to have a personalized treatment that you could offer a patient at that point.
Are there any treatments that are being considered for the dry form of AMD?
There are trials that are in the works. Our group has had exciting but very preliminary data. We're using high-dose atorvastatin or high-dose Lipitor for a certain group with dry macular degeneration. And in that small study that was led by Demetrios Vavas and a collaborator in Crete, Dr. Silimbaris, we were able to show that we could really eliminate these large drusen deposits with preservation of the retina and preservation of vision. And so we're hoping to get to be able to do a larger trial to really answer the question as to whether that works.
We're excited to see there's research being done on minimizing progression of AMD. Is there anything patients at risk can do to prevent the progression of AMD currently?
I mean, the education is still helpful to patients, but you're right, we don't have a drug treatment to offer and really are limited to saying it's lifestyle changes, so very heart-healthy diet, don't smoke, and we're indicated to take vitamins based on the ARAD studies.
Would you like to share some information about the AMD symposium?
So this is our fifth symposium. We run this every other year, so we started 10 years ago. And the motivation was really to have a symposium where you could bring in experts investigating macular degeneration, but also experts from other disciplines that could really help us tease apart and improve our understanding of macular degeneration. So we have continued to do that, and it's been very successful. We've had experts from the fields of lipids, cardiovascular disease. Today, our metabolomics expert really works in asthma. We have experts, Andrew Dick, this time also from working in inflammation, not always directly in macular degeneration. And that kind of cross-pollination, I think, really helps drive the field forward. We also make sure there's a lot of time for discussion and networking, and I think that ends up making the meeting a lot of fun for the participants.